Hey everyone, in this video we are continuing sharing information about spike proteins and clotting, these unusual clottings that are being observed post-COVID-19. And in today's video, we're going to be looking at first images of what these clots look like under the microscope based on what's been published in scientific literature. And my name is Dr. Mikola Rashek of Neurogenomics, and let's get started. So the group of people that are publishing this information, they are clotting experts from the leading author, I believe, is from South Africa, and they've done a lot of fantastic analysis of this information. That's where the bulk of my knowledge comes from, is from this particular group. And in the two of their papers that I'll be discussing in today's video, they did a couple things. So they did protein analysis. That can also be referred to as proteomics. Basically, this is where you're looking at composition of proteins in a given sample. We discussed this briefly in our series dedicated to exosomes, to determine what kind of proteins exosomes could be carrying. And remember, exosomes were tiny fragments of cells, and the series was dedicated to, is it possible that exosomes carrying spikes could be breathed out? And basically that would constitute a form of shedding, right? So that's when we introduce the concept of protein analysis already. And the other thing that they did is they did fluorescent microscopy. So fluorescent microscopy, we've also discussed this already many times. Again, in exosome series, we discussed that a lot, as well as some other notable example would be we introduced fluorescent microscopy also when we were discussing the possibility whether the spike protein could be entering the nucleus. There's few videos dedicated to that topic as well. And in essence, what you're doing here is you are labeling antibody with certain fluorescent tag molecule, and then that you use antibody to target protein of interest, and that allows you to see where that protein might be localized. And the authors did something similar in this case, but instead of having a fluorescent molecule attached to antibody, they did something else. They used a fluorescent molecule known to only interact with amyloid structures. So what does that mean? We're going to expand on this topic a lot more in the next video. Amyloid structures are basically unusual forms of protein structures that are not normal, but they can be triggered, and they adopt specific structure that this specific molecule called thioflavin T can interact with, and fluoresce and start fluorescing once you hit it with a specific light wavelength. And in this case, you can basically see where you have amyloid structures, so these proteins that are not properly structured, and that's one of the ways of studying unusual protein structures. Basically, they're misformed, if you will, and this is one of the hallmarks of these clots that are being uncovered right now, is that they're not like normal clots that we observe during normal natural clotting process to help fight the damage to our vascular system. These clots are abnormal, and having this unusual amyloid structures is quite significant, as we will see right away in a moment. Okay, so let's maybe start showing these images. So thioflavin T molecule exposed to blood samples, and then thioflavin T will go and interact with these improperly folded clots, and it shows you basically where there are clots. And what's interesting in the very first image is that in that very first image, they, in panel A, you can see they had this is neat because they had a comparison. They had a blood sample of an individual prior to getting COVID-19, so prior to pandemic, and then that very same individual also got long COVID, and they were able to show there's a difference that post long COVID, that individual was showing greater amount of these fluorescent blobs. Now, if you look at the size of these, they're big, and there was one big one even prior to COVID-19. If I had to guess, it means that this particular individual would have had certain predisposition maybe to clotting anyway, but you can clearly see that the level of clots are enhanced. So what you're looking at really is these green fluorescence, and that green fluorescence shows you basically where the clot is and its structure. It's even its three-dimensional patterns. And then the next example below is basically a bunch of examples of long COVID patients and the amount of clotting that can be observed in their samples. And you can see maybe like what the take-home message here is that there's a variety of patterns. So you can see those little dots. So these clots are can be super tiny. And if you wonder the size, if you look at the very left-hand bottom of these images, you can see there's a little white bar, and that's the size that it tells you that it corresponds on the image. So that tiny little bar measures approximately 10 micrometers. If you wonder what kind of unit of length that is, 1 millimeter is made up of 1,000 micrometers. Right, so 10 micrometers is basically 1/100th of a millimeter. And you can see in some of these images, it's a bunch of little dots. They look like tiny little stars in the sky, right? So you can clearly see that there is lots of clots. But then in other images, you can also clearly see that these clots can be bigger in size. So perhaps they can assemble into bigger sizes. Not sure whether it's these small clots that can then continue to coalesce together to make these bigger ones. That's my assumption. And you can see that they can get quite big in size. Most of them are round, but then even in this particular example, you can also see that you can have like tube-like or worm-like like the bug structures, like these lengthy ones, and they can be huge in size. Like if you start looking at this, they can be a few hundred micrometers in size, and some of them don't even fit in the entire image. So they obviously means they're bigger. And we've seen some of these uncorroborated images online of embalmers pulling out these unusual clots. I still have not found any scientific analysis of that information based on cadavers at all. So in terms of science, doesn't exist. Therefore, not something I want to comment on until I guess some scientists start maybe analyzing this, if it's real, right? Because that's what we need to know. Is this even real? But you can see that you already have these long tubes forming as well. So definitely unusual. And you can even see in one of the images, it's like a tube with a hole in it, right? So what the authors did next, this is really neat, is they exposed these samples to trypsin. Now, trypsin is like a molecule that can destroy proteins. It cuts them up. It's like basically molecular scissors that will just start chewing up proteins into fragments. So you can destroy these. This is called trypsinization process. And normally, if you take a normal clot and expose it to this trypsinization process, it should destroy the clot. And indeed, this is what they observed. This is what they observed with clot samples from healthy controls, meaning healthy individuals that were used as controls, as well as individuals with type 2 diabetes. And in this next image, you can see that after trypsinization, the controls and type 2 diabetes, there's basically nothing there. There's a couple white arrows pointing to like super tiny remaining fragments, but pretty much everything was destroyed in the process, and you no longer have fluorescence because that molecule, thioflavin T, has nothing to bind anymore. But when it comes to acute COVID individuals or individuals with long COVID, you can still see lots and lots of clots remaining. What it means is basically trypsinization. One round of trypsinization was not capable of destroying these clots. That's how resistant they are to degradation. And this is why these clots formation post-COVID, and we'll talk about this in soon coming up in future videos, that spike protein could have big influencing factor here to generate these abnormal clots. But because these clots are not normal structure and they are more resistant to being degraded, they can persist much longer than what would be otherwise expected. Okay, so clearly you can see in individuals with long COVID or acute COVID, these clots continue to persist. So not a good thing. And again, you can still see the variety of clotting flavors, if you will, if I can say like that. So you see these tiny points. Again, I call it like, it looks like stars in the sky. And then you have these big rounded ones. And to continue with analogy, I call these like supernovas. And then you have these long snakes as well. So again, these clots clearly can get to very large size, and that cannot be a good thing to a person to have something of that size floating about in the blood. There's one more image from this particular paper, just simply more examples of long COVID individuals or acute COVID individuals and their clots. And this last image I found particularly interesting because the resolution is very nice, because you can actually see this more detailed three-dimensional patterns as the dye intercalates into these misfolded structures. You can see how unusual they can be. They can take unusual three-dimensional forms, and once again you see some that are massive. They literally look like they're a few hundred micrometers in size. So big, really big. Not a good thing. So those two elements of information, the fact that these clots can stain with this thioflavin T, which normally stains amyloid structures. So these structures that when they form, they can cause diseases. There's many of these known, many different proteins known that they can achieve that. So Alzheimer's is one example. Prions in mad cow disease is another example, right? And so that's one clear point of evidence showing that there's something is strange about these clots. And the other one is that they're also resistant to destruction. So in the combination of the clots being triggered into formation, as well as clots not being broken down properly, it means that such clots could persist for a long time. Their persistence in the circulation could easily mean that they could possibly start blocking certain blood vessels, And the authors believe that this would lead to hypoxia, where you no longer have oxygen delivery, proper oxygen delivery to tissues. So there's not enough oxygen and carbon dioxide exchange in these tissues. And they believe this is likely the #1 reason explaining the vast majority of the complications that are being seen with these clots. So that's enough about the images. Remember, I also mentioned that they did proteomics, so they did protein analysis. So they wanted to see what is found inside those clots. So obviously fibrin is expected to be there, and that's exactly what they saw. Recall in the previous video, I provided a summary review of how clotting normally occurs, and that fibrin is the central component in making clots. So not a surprise they found that, but they found other proteins, other molecules as well in this first round of analysis. And one of the proteins they found was that alpha-2 antiplasmin, which is an inhibitor of plasmin, and plasmin is also central into degradation of clots. So what the authors are suggesting is that perhaps these clots entrap molecules that also might reduce the likelihood of their degradation as well, because of the type of molecules that they might entrap. And the other molecule in this particular study that they were finding was, I believe it was called serum amyloid A, and it's a family of proteins that can participate in the activation of the immune system, both the innate and adaptive immune system. So remember, there's two different components of immune systems. One that is always present, that's innate, and you don't need to trigger it. And then adaptive, that's the one that you need to trigger into existence by exposure to the pathogen. And the authors mentioned that it's been known that the level of this protein, the higher the level of this particular protein in the blood, correlates with the severity and then mortality of COVID-19 as well. So, and that apparently was also found inside those unusual clots. Now, they did another study where they analyzed protein makeup as well, but they had larger sample size. So well over 100 individuals that they were looking at, and once again to see what's inside there. And they found increased levels of that alpha-2 antiplasmin, but they also saw some other proteins as well. So one of them was von Willebrand factor. So what is that? That's a molecule that attracts platelets, and it basically helps activated platelets to start assembling at the site of injury. So they clearly contribute to clotting as well. Also, they found platelet factor 4. That's one of the molecules that is released by activated platelets. And on this topic as well, I should also mention the authors also showed that platelets are hyperactivated in long COVID individuals. So what does that mean? Is that there's a lot of them being activated in both acute COVID, but even more so in long COVID. And they did show images of that as well, where they used, again, fluorescent microscopy, and they targeted two molecules that are found on the surface of the activated platelets, and they're found there when the platelets are activated. One of the fluorescent molecules was purple, the other one was green. Basically, both of them can be found at the same time, and you can clearly see in controls, you can have some activated platelets, and then you see these cute little purple/greenish dots. And that basically shows you where the platelets, activated platelets, are in the blood. And as you can see, that people with acute COVID-19, they have a lot more of these. But what's also interesting with long COVID is that not only do you have more of these, they also can assemble, aggregate together into large masses. So why is that not a good thing? This is because highly activated platelets, when you activate platelets, they release a lot of molecules, including that platelet factor 4 I just mentioned, but they also release a lot of molecules that promote inflammation, and that in itself is also dangerous. Such dysregulation of activation of platelets can even lead to inflammation diseases if it's not properly balanced. So not necessarily a good thing. So what else they found inside those clots? There was another protein. Trying to remember what the name was. I don't even know how to pronounce the name. Something like kallikrein. Another name for it is KLKB1. Now that one is an interesting molecule because it actually helps to also convert plasminogen to plasmin, which is the molecule you need to start clearing up clots. So that one normally is good for what you want in your circulation, but this is entrapped in these clots as well. So you can see they found a number of different molecules, and they also found antibodies entrapped in these clots as well. Also interesting, although I wish we could have known what the nature of these antibodies are as well. That would have been interesting to find out, and I believe the authors were indeed investigating that, or they are in the process of investigating this. So I definitely will be looking forward to that information as it comes out. So there you go. This is what these clots can have in them. And on the topic of clots, the authors also mentioned that because these proteins are being trapped inside these clots, what might be happening is that normally when you're looking for markers of what is happening with the blood, for these inflammatory markers, you look at them at floating in the blood, and as a consequence, these changes might not be detectable because these molecules are entrapped inside. They're entrapped inside these clots. So you might not have capacity to actually properly measure these changes. So that's something basically to watch out for. So that's it for this particular video. In the next video on this series, we're going to talk about in greater detail about amyloidosis. So we're going to break that down a bit, and then we're going to follow that up with the ones that I'm really looking forward to, and showing you and explaining is basically the role of spike protein in the entire process. And there has been some amazing science already done by multiple different groups showing how spike protein is influential in this entire process. All right, my dear friends, this is it. Thank you again for supporting the channel. Please give us a like. Please share the video. Please join my Patreon account. That's slowly growing, so that's wonderful. There's different types of content goes into those videos. This is the type of content that you were asking for as well, but we didn't have a really good opportunity to produce it for this channel. So this is basically the content that people have been asking in some of the questions we've been receiving, and we're finally have a venue for being able to answer some of these. So more controversial stuff will end up in there, okay? But still always based on science. So the format is same in that I basically always talk about what kind of scientific literature I came across, basically, right? And once again, please check out our COVID-19 Q&A events. This is basically if you want to get a free ticket to that, subscribe to the newsletter, and we'll send you a free ticket. Otherwise, it's a Q&A. First, we answer a few questions there, and then it's open mic. And typically, what happens is the audience influences basically some of the material that I study, because I really, really appreciate this, because different audience members bring different scientific papers for discussion, and some of them make it to these videos. There's just too much information to make video on everything. Luckily, other really, really good channels fill in the gaps that I just would not be able to produce all of this content. So, but thank you for the support again, and thank you for being with us for so long already. So I'll see you folks in the next installment on the series dedicated to defining what amyloidosis is all about and how that plays with regards to these clots formation. Bye everyone, ciao for now.